what I'm going to be talking today about is the BioAge platform, which is deeply rooted in human omics data, which we use to generate biomarkers, which also shine a light on mechanisms and have taken us now into drug trials, into therapeutic trials within mechanisms of aging, and which we are about to be able to then generate molecular data from those trials to take us back to biomarkers. And that's what I'm going to walk you through today. All right, so BioAge, we are a platform-driven clinical stage biotechnology company. We're advancing a growing portfolio of therapeutics that treat severe diseases, but they do that by targeting root causes of aging. We have a powerful human-first discovery platform based on the kinds of human data that I was talking about. Our clinical stage portfolio has 3 programs and growing. And of course, we have proven track record of results and have raised some money from venture capital to support us. So at BioAge, like many of the people here at the conference, we would love to change the nature of aging, and this is an inspirational question. What could you do with more healthy time for you and your loved ones? And we think about that a lot. Clearly, aging is a huge cause of morbidity and mortality. And, in fact, addressing any of these individual diseases on the left will not have a huge impact on health span or lifespan because it won't affect the trajectory of any of those other diseases. So really, there's huge promise in trying to address these underlying mechanisms of aging to extend human health span and lifespan. And clearly, there are these awesome examples in nature that teach us that this is not an effort that we're pursuing in vain. There is no hard limit on the lifespan of an organism, certainly not at 80 or 100 years. We have Greenland sharks living up to maybe 400 years or more. We have bristlecone pines with many millennia of history to them. So that tells us, this is a potentially tractable problem. And even better than that, there's been a lot of really great work in recent decades demonstrating that not only is that a theoretical possibility for organisms to be able to modulate lifespan, but it can actually be done. And so in a number of model organisms, including, Drosophila, C. elegans, and now, mammals, including mice, it's been reproducibly shown that you can intervene, sometimes at sort of, birth or pre-life and sometimes in the middle of life to extend health span and lifespan. There's been, numerous genetic interventions in invertebrates that really have stunning impacts on lifespan. In mice, there's interventions like rapamycin, senescent cell ablation that can do this. And that's really, inspirational that we can do this for humans too. And it's really only in recent decades that we've begun to develop the tools to seriously study aging in humans, which at BioAge, we strongly believe in that if you want to make therapeutics that will successfully modulate aging in humans, that you have to be discovering the right translational mechanisms that will work in humans using human data. And so it's really, in recent years that we've been developing robust capacity to use multiomics to interrogate the biological process of aging in humans. And what we do at BioAge is apply that to really the natural human experiment that's happening all around us. And in particular, it's happening in our healthy aging cohorts, our BioAge healthy aging cohorts. And it's this, distribution of trajectories of health span and lifespan. So clearly there are these stunning examples on the right here, like Robert Marchand, who set a record as the oldest competitive cyclist, age 105. That he was even a competitive cyclist at all at 105 is stunning. And he lived to be 109. And then, there's people like that. And then on the other end of the spectrum is, people like the rest of us who begin to get our chronic diseases in our 60s, and many of us die in our 70s and 80s. And, for I think the majority of us who, I'm not a competitive cyclist, but for those of us who are competitive cyclists, probably, most of us wouldn't do it much past our 40s or 50s. So there's this, really striking difference in health span and lifespan. And the question we ask at BioAge is what are the molecular underpinnings of these different trajectories? We can take samples from people in healthy middle age when all of them, look basically the same from the outside, and really figure out the biological differences that will ultimately lead some of those people to have, a fast aging trajectory, with more unhealthy and leads to quicker death, versus this really robust trajectory. We are very much data-driven at BioAge. So we use AI and multiomics to really directly decode the biomarkers of aging and also the underlying human biology that's driving aging. And so we collect data, as I was saying, over the lifetime of people by partnering with healthy aging cohorts, where people were tracked over decades and had samples collected, which we can interrogate using modern multiomics technologies like proteomics, metabolomics, et cetera. And so our platform to date has assembled many tens of millions of molecular data points, many thousands of patients, and follow up to many decades there, up to 45 or more years. And that really lets us ask the question I framed earlier of what's the biology that separates the people who age well and who age poorly. And a really key feature that lets us do this is being able to measure the phenotypes directly in humans on these cohort participants that really tell us whether they're aging well or poorly. And on the left, of course, a key one here is lifespan. Lifespan is a really key feature of aging. It's really everything that goes wrong with you as you age sort of dovetails in your ultimate risk of dying. So, so that's a key feature. But also on the right, these phenotypes that really tell us about how well you're aging, how functional you are, how healthy. And that's things like cognition, like cardiometabolic status, like muscle function on the bottom. Your grip strength and your walking speed and your mobility really tell us a lot about whether you're a healthy, active person who can do the things you want to do, or whether you're, very limited and potentially, confined to your house or an institution. And so these phenotypes let us ask, and again, in a data-driven way, which mechanisms are leading to people who are, A, long-lived, but B, really healthy and functional. And this is just a great illustration of, the data-driven aspect of looking for biomarkers and mechanisms in untargeted omic data. So you can see illustrated here in the center, just a plot of around 6,000 proteins and their relationships to all-cause mortality on the y-axis. The x-axis is meaningless here, it's just for illustration. And the scale of these circles also scales up with the effect on lifespan. And then you can see highlighted in different colors some associations with key aging phenotypes. So picking out the individual proteins that not only have an interesting association with lifespan, but also have an association with one or more of these specific health span features that we picked out here, like muscle aging, brain aging, renal aging, cardiovascular aging. And so this landscape lets you see exactly how these signals can come out of the data. And this is essentially the raw data that feeds into our platform, which we use to discover the key relationships, biomarker and functional relationships for aging. And we not only look at the associations with outcomes, we also look at the molecular architecture, at what the cohort data is telling us about how these molecules like protein, the metabolites, and transcripts relate to each other in aging individuals. And so you can see this progression on the left, where we start with pretty much an initial map of some of these molecule-molecule interactions. We can then begin to cluster them into functionally related groups. And then in the third panel, we can prune out the relationships that are more likely to be spurious, less likely to be causal or functional. And then finally on the right, we zoom way out and flatten everything to see kind of an illustration of one piece of the grand architecture here of what we've derived for functionally related groups in these aging individuals. And despite the fact that these groups were derived only based on the molecule-molecule interactions and not on any of these clinical phenotypes, each of these clusters has shown really interesting associations with specific aspects of aging and function with age, excuse me. And I think you can begin to see from those two slides I was presenting how this data-driven approach can really help us illuminate biomarkers and also mechanisms in aging biology. And so you can see in the middle illustrated, the wheel of the hallmarks of aging, which is really a landmark sort of way of thinking about the different mechanisms and organizing them. And many of the mechanisms that we know that fit into those categories have, of course, been established in model organisms. And one thing this data-driven approach can do is really shine a light on these known mechanisms, on these known categories, and tell us which of these really show strong signals in humans are much more likely to translate to therapeutics in humans and which of them aren't. But not only that, it can give us a peek at what we've playfully called on here on the right, the untracked wilds. So places where the literature hasn't gone yet. We just haven't been able to discover using our existing approaches. And because we're letting the data tell us about what pathways associate with aging in humans, we can also have the chance to find completely novel and powerful mechanisms that are also operating in aging. At BioAge, we focus on a few thematic areas for our own internal drug development, and that's immune aging, muscle aging, and brain aging. And today we're focusing on muscle aging for some key examples. So I'm going to tell you the story of how one of our drug programs, which relates to apelin, which we'll talk more about, came out of the platform and what's happened to it. So interest of time, I'm not going to go over everything in these slides, but given that we're really interested in muscle aging, one thing we did was worked in our platform to generate a muscle aging metric on the bottom there, which incorporated A, longevity, but B, all the sort of relevant aspects of muscle function and functional ability in people that relates to muscle function. And that's right there, the histogram of, in this case, more than 5,000 proteins that illustrates each of their associations, or sorry, each of their scores on this metric. And that tail there is the most, on the right, where apelin is labeled as the most interesting proteins. Apelin is one of the ones that's in that tail. And interestingly, it was, the most interesting protein in that tail that had an asset that existed in pharma that we could potentially in-license. And when we delved a little bit more deeply into the data, we saw this really, compelling relationship on the right between apelin protein levels in people, and in blue, the chance of really living a long time, and in purple, the chance of living a long time with preserved grip strength. And there's a nice, essentially dose-response relationship there. We also kind of zoomed out to that network view where I talked about, how we generate the architecture from these aging cohorts. And so looking at the broader pathway surrounding this molecule also was really interesting. And one example here on the right is how the network level activation also correlated strongly and significantly with all-cause mortality, which is the hazard ratio there on the y-axis. The last sort of step in our process of using molecular data and biomarkers and proprietary human cohorts to identify targets that we want to move forward with is to do experimental validation in animals. And we proceeded to do that. I'll tell you a bit more about it. So I'll skip over later. So I'll skip over this bottom part here now. After having seen these great signals from the cohort and gotten our interventional data, actually using some molecule that we borrowed from Amgen, we decided to in-license Amgen's molecule, which is an APJ receptor agonist. So it basically activates the apelin pathway, but it's a small molecule instead of being apelin. And briefly, it's an oral, exists in oral form and an IV form, clean safety profile in a prior phase 1. And we've now initiated a phase 1b trial in healthy elderly volunteers at bed rest, where we're looking at safety and some exploratory endpoints related to muscle function. I'll tell you a bit more about that in a second. But apelin is really interesting because it was previously known in animals. There've been a couple of great studies showing that apelin is secreted by muscle, particularly active muscle, and that with age, the activation of the pathway goes down, but that adding apelin to aged animals could rescue some aspects of their physical function. And again, this is an example of how it's really critical to be able to take some mechanisms that work in animals and be able to distinguish which of them really are likely to be, or potentially likely to be, translational to human therapeutics. And so now this is some of our animal data here supporting that. So on the left, we put a bunch of old mice on running wheels and watched them age out another 80 days. And you can see that the treated mice preserved their physical activity, their running wheel activity over time, as the curves really are significantly separating over time. On the right, you can see the before and after for those same mice for grid hanging times. And you can see that while mice that received vehicle had substantially decreased grid hanging times, that mice who got the BG105 did substantially better. You see not nearly as many mice that have massive drops, and you see these mice that didn't exist before that actually got better. We also did a casting study where we casted mouse legs, and when they're casted, they can't move that leg, and so the muscles atrophy with time. And we gave them drug to see if that would help. And you can see in the uncasted limb on the right that there's really no difference. But that in the casted limb on the left, you can see that a lot of atrophy was happening in the vehicle-treated mice, but basically no atrophy at all in the BG105 treated mice. So this was really gratifying data package, and that led us to be interested in taking a look at this drug for muscle atrophy in older individuals. And so right now we're running a test that, as I said, is looking at safety, PK, biomarkers, of course, proteomics, and also starting to look at some exploratory endpoints for muscle to see what older adults who are at bed rest, who again then are inactive and experiencing atrophy, what this drug can do related to their muscle function. And the bigger context there is that what we really like to do at BioAge is take a promising aging drug forward first for a model indication. In this case, something related to atrophy. For example, ICU diaphragm atrophy is a great one there. And then use that as a stepping stone to get to bigger indications that are real broad, sort of inspirational aging indications, like in this case, sarcopenia and frailty. And so that's where we see the short, medium, and long-term value of this drug. And now getting, we went from biomarkers to drug trials and how we go back to biomarkers. And that's this great way in which, when we do drug trials, we take blood samples from these patients, which we run the same omics modalities on. And this allows us to feed back into the platform in a lot of useful ways. Not only does it expand the platform with more elderly individuals, but it allows us to interrogate very specific effects on human biology of the drug. Can we confirm existing ideas about the mechanism? Can we learn new things that weren't known before about the mechanism? It allows us to look specifically at aging-related impacts. Are any of those hallmarks affected, by this drug, or any specific known aging pathways? And does this drug affect known aging biomarkers? Does it, increase your, biological age or decrease it? And last, new clinical biomarkers that are really important for clinical development. Can we, derive signatures that are important for selecting patients who are likely to have a better effect from the drug? Or can we use them to identify when a patient is starting to have an effect for a drug really early on? And so this is a great way that we derive a lot of value from the clinical trials, not only with the main clinical outcome, but also with the biomarkers that feed back into the platform. And I'll just end with this great quote from Tenley Albright, a surgeon, which parallels our philosophy. Our aim should be to help our patients die young as late as possible.